In September of 2023, a small capsule the size of a kitchen pot slammed into the Utah desert at roughly 36,000 kilometers per hour. It had traveled nearly 7 billion kilometers to get there. And inside it, sealed in a container smaller than a shoebox, was material that had not touched the Earth or any planet in 4.5 billion years. That number deserves a moment. 4.5 billion years. If you compress the entire history of Earth into a single calendar year, modern humans appear in the last 14 seconds of December 31st. The material inside that capsule formed before the first second of January 1st, before the oceans, before the continents, before the first single-celled organism drew what we might loosely call a breath. This was not a rock from the ancient past. This was a fragment of the raw, unprocessed material from which the entire solar system, the sun, every planet, every moon, every ocean on Earth, and every atom in your body was assembled. The mission was called OSIRIS-REx. The asteroid was called Bennu. NASA engineers were not casual about this. The return capsule was designed with multiple redundancies. A parachute system slowed its descent. A recovery team had been stationed in the Utah Test and Training Range for weeks. Helicopters tracked the capsule through the atmosphere on its final approach. When it landed, technicians in specialized cleanroom suits retrieved it within hours, sealing it in nitrogen-purged containers to prevent even a single molecule of terrestrial air from contaminating what was inside. The level of caution was not theatrical, it was scientific necessity. Because what Bennu's surface contained could answer questions that no laboratory on Earth had been able to answer through any other means. Here is the question at the core of this story. Where did the building blocks of life come from? Not life itself. That remains one of science's open frontiers. But the chemical architecture of life, the amino acids that form proteins, the nucleobases that encode genetic information, the organic molecules that allow chemistry to cross the threshold from mere reaction to biological function. These molecules exist on Earth. They are in you right now as you listen to this. But did they originate here? Or did they arrive from somewhere else, carried by objects exactly like Bennu, billions of years ago, during a period so violent and so ancient that the geological record of Earth has almost entirely erased it? The OSIRIS-REx mission was designed in part to test that hypothesis with real material, not meteorites that had been contaminated by Earth's atmosphere and biosphere during an uncontrolled fall, but pristine, carefully collected, hermetically sealed, surface samples from a carbon-rich asteroid that had been drifting through the solar system, largely unchanged since the beginning. Bennu is not small. At roughly 500 meters in diameter, about as wide as the Empire State Building is tall, it is large enough that a collision with Earth would release energy equivalent to roughly 1,200 megatons of TNT. For reference, the largest nuclear weapon ever detonated by humanity, the Soviet Tsar Bomba in 1961, released approximately 50 megatons. Bennu would be 24 of those, in a single impact. Bennu is classified as a potentially hazardous asteroid, and current orbital models give it roughly a 1 in 1,750 chance of impacting Earth sometime between 2178 and 2290. That probability is not zero. Scientists watch it carefully. But Bennu's potential danger is not what drew NASA to it. What drew scientists to Bennu was its composition. It belongs to a class called carbonaceous chondrites, or more specifically, it is classified as a B-type asteroid, one of the most primitive and chemically unaltered types known. While most rocky bodies in the solar system have been transformed by heat, pressure, volcanic activity, or collision, Bennu has experienced relatively little of that processing. It is, in the language of planetary science, a primordial remnant, a time capsule. Its surface and interior preserve chemistry from the solar nebula, the vast disk of gas and dust that surrounded the young sun before any planets had yet formed. The OSIRIS-REx spacecraft launched in September 2016. It took over 2 years to reach Bennu, arriving in December 2018. For nearly 2 years after that, the spacecraft mapped Bennu's surface in extraordinary detail, imaging it at resolutions as fine as 5 centimeters per pixel. The surface was nothing like scientists had predicted. Models built before the mission assumed Bennu's surface would be relatively smooth, a fine-grained regolith of dust and small pebbles, easy to collect from. What OSIRIS-REx found was a rugged, boulder-strewn landscape. Boulders the size of multi-story buildings jutted from its surface at irregular angles. The terrain was chaotic, angular, far more complex than anticipated. Mission controllers had to redesign the sample collection approach in real time, identifying a small, relatively clear patch in a crater called Nightingale, a depression near the north pole of the asteroid, just 16 meters across. To collect the sample, the spacecraft did not land. It could not. Bennu's gravity is roughly 100,000 times weaker than Earth's. An object that weighs 100 kilograms on Earth would weigh approximately 1 gram on Bennu's surface. The gravitational pull was so weak that the gas thrusters used to stabilize OSIRIS-REx during collection could have launched loose material into orbit around the asteroid. Instead, the spacecraft performed what is called a touch-and-go sample collection. It descended slowly on a planned trajectory, extended a robotic arm tipped with a specialized collector head, made contact with the surface for approximately 6 seconds, fired a burst of pressurized nitrogen gas to stir up and capture surface material, and then fired its thrusters to ascend immediately. 6 seconds of contact. After a journey of nearly 7 billion kilometers and 4 years in space. When the collector head was inspected by the spacecraft's cameras after the event, engineers immediately knew something extraordinary had happened. The head was so full of material, an abundance they had not expected, that the mechanism designed to seal the sample container was being partially blocked by rocks jammed in the closure. Material was visibly leaking from the collection device. They had collected too much. Scientists had hoped for at minimum 60 grams. The eventual measurement confirmed they had collected approximately 250 grams, more than 4 times the mission's minimum requirement. That sample landed in Utah. It was rushed to a specially constructed clean room at NASA's Johnson Space Center in Houston, Texas, where a team of scientists from institutions around the world had spent years preparing to analyze it under conditions that matched, as closely as possible, the vacuum of space itself. What they found inside broke expectations immediately. Not in the way of catastrophe or crisis, but in the way that only genuine science can break expectations, quietly, methodically, and with enormous consequence. The early analysis reports noted the presence of hydrated silicates, minerals that form only in the presence of liquid water. Bennu, or more precisely the parent body from which Bennu's material originated, had once interacted with water, not a passing encounter. A sustained chemical interaction. Water had flowed through this rock, or through the rock that became this rock, in the early solar system, billions of years before Earth's oceans formed. And then came the organic molecules, carbon-bearing compounds, complex ones, the kind that, in certain arrangements, begin to look like the molecular precursors to biology. The scientific community did not panic. It did not celebrate prematurely. What it did was go quiet and careful and begin to ask a question that is among the most profound in all of science. Not what is here, but what does it mean that it is here, and how did it get here? And could this same material have arrived on a young, wet Earth 4 billion years ago, seeding the chemistry that eventually looked up at the stars? That question does not have a complete answer yet, but the search for it changed everything scientists thought they understood about where life begins. 121.6 grams. That is the total mass of the Bennu sample that reached the clean rooms of NASA's Johnson Space Center in Houston. To hold that number in your mind, take a standard stick of butter from a kitchen refrigerator. It weighs 113 grams. The largest asteroid sample ever returned to Earth by any spacecraft in human history weighs slightly more than a stick of butter. And yet what those 121 grams contained redrew the map of how scientists think about the origin of life on this planet. The first published analyses came in 2 waves. The initial findings arrived in October 2023, weeks after the capsule landed. Then, in January 2025, the first in-depth peer-reviewed results appeared simultaneously in the journals Nature and Nature Astronomy, 2 of the most rigorously reviewed scientific publications on Earth. Authored by scientists from NASA and research institutions across multiple countries. The findings were not preliminary impressions. They were the product of more than a year of methodical, controlled laboratory work conducted on a sample that had been protected from terrestrial contamination at every single step. The opening finding was already remarkable. The sample was nearly 5% carbon by weight. 5% sounds modest until you consider what it means at the scale of an asteroid. Bennu has a mass of approximately 73 billion kilograms. 5% of that mass, expressed as carbon, represents more carbon in a single asteroid than exists in all the coal reserves currently mapped in the United States. Carbon is not exotic. It is the fourth most abundant element in the universe. But finding it in this concentration, in this form, in material this old and this unaltered, confirmed what scientists had long suspected about carbonaceous asteroids and hadn't been able to prove with pristine, uncontaminated samples until now. Alongside the carbon came water. Not liquid water, not standing pools or flowing streams inside a rock, but the chemical signature of water that had once been there and had permanently restructured the mineral architecture of the asteroid. The sample was saturated with hydrated clay minerals, specifically phyllosilicates, which are layered silicate structures that only form when liquid water interacts with rock over extended timescales. On Earth, phyllosilicates are found in riverbeds, in ocean sediments, in the geological layers laid down over millions of years of wet geological history. Finding them in Bennu means that at some point, the parent body from which Bennu originated, a much larger object that was shattered by a collision billions of years ago, experienced prolonged, sustained contact with liquid water. The pH of the water that had once moved through that material was measured at 8.23, slightly alkaline. That is not a random data point. Weakly alkaline aqueous environments are, according to current models of prebiotic chemistry, among the most chemically productive environments for the synthesis of complex organic molecules. The ocean floor hydrothermal vents that many scientists believe played a role in the origin of life on Earth operate in conditions broadly comparable in their chemistry. The rock from Bennu was not just wet. It was wet in a way that was chemically productive. What followed from that water, what the water made possible, is where the January 2025 findings crossed from interesting into genuinely staggering. 14 of the 20 amino acids that life on Earth uses to construct proteins were detected in the Bennu sample. 14 out of 20. Amino acids are the molecular units that link together in long chains to form proteins, and proteins perform nearly every structural and biochemical function in every living organism on Earth. They fold into enzymes that drive chemical reactions. They form the structural scaffolding of cells. They carry oxygen through blood. They transmit signals between neurons. Without amino acids, there is no protein. Without protein, there is no biology as we know it. And 14 of the 20 that life on Earth relies upon were sitting inside a piece of rock that formed 4.5 billion years ago, more than 2.5 billion kilometers from the sun, long before Earth had oceans, long before Earth had continents, long before Earth had anything. The mixture was notable not just in variety, but in its symmetry. Amino acids can exist in two structural forms. Like left and right hands, they are mirror images of each other, called L forms and D forms. Life on Earth uses almost exclusively L-form amino acids. When amino acids form in purely chemical processes in space, without any biological influence, they tend to produce equal amounts of both forms. The Bennu sample showed a racemic mixture, roughly equal quantities of both L and D forms, which is precisely what you would expect from non-biological abiotic synthesis. This was important. It was scientific confirmation that these amino acids had not been produced by any biological contamination on Earth. They were genuinely extraterrestrial. They had assembled themselves through chemistry alone, in the cold and the dark of space, driven by nothing more than the physics of molecular interaction and the energy of the early solar system. Alongside the amino acids, researchers detected all 5 nucleobases that life on Earth uses to encode and transmit genetic information. 5 out of 5. Nucleobases are the information-carrying molecules in DNA and RNA. Their names, adenine, guanine, cytosine, thymine, and uracil, are not the language of science fiction. They are the actual alphabet of heredity. DNA is essentially a long chain of instructions written in a 4-letter code built from these 5 molecules, and RNA transcribes those instructions and uses them to assemble proteins. Every organism that has ever lived on this planet, every bacterium, every fungus, every plant, every animal, stores its biological blueprints using these 5 molecules. And all 5 of them were in the asteroid. What was not in the asteroid, to be precise, is equally important to understand. Nucleobases are not DNA. Amino acids are not proteins. These molecules are ingredients, not products. Finding them in Bennu is not the same as finding life, or even the direct precursors of life in their assembled form. What it shows is that the raw chemical toolkit, the molecular hardware that life on Earth eventually selected and organized into biology, was being manufactured in space, at scale, billions of years before life appeared on this planet. This brings up a myth that deserves direct dismantling. There is a widespread assumption in popular culture that the origin of life is synonymous with the origin of Earth, that somehow the molecules needed for biology spontaneously generated from scratch on our planet, independently, from a cold, barren, purely mineral surface. This assumption has been weakening for decades in the scientific literature, and the Bennu results strike it directly. The solar system, during its formation and early history, was an extraordinarily chemically active environment. The same processes of aqueous alteration, ultraviolet irradiation, and organic synthesis that occurred inside Bennu's parent body were occurring simultaneously in hundreds of thousands of similar carbonaceous bodies throughout the early solar system. The solar nebula, the cloud of gas, dust, and ice from which everything formed, already contained complex organic molecules inherited from interstellar space, carried in the mantles of dust grains that predated the sun itself. Life on Earth did not necessarily invent its own molecular toolkit. It may have inherited it. The delivery mechanism for that inheritance is called the Late Heavy Bombardment, a period roughly 3.8 to 4 billion years ago, during which the inner solar system, including Earth, was subjected to an extraordinary influx of asteroid and comet impacts. The geological record of this period is almost entirely erased from Earth's surface by billions of years of tectonic activity and erosion, but it is preserved on the lunar surface in the form of ancient impact basins visible today. During that bombardment, carbonaceous asteroids chemically similar to Bennu were striking the young Earth with regularity. They were delivering water. They were delivering carbon. And if the Bennu data is representative, which current evidence strongly suggests it is, they were delivering amino acids, nucleobases, ammonia, and sugars, not in trace quantities, in abundance. Speaking of sugars, a study published in Nature in December 2025 detected bioessential sugars in the Bennu samples. Ribose and related sugar molecules, the structural backbone of RNA, were present in the material. RNA is believed by many origin-of-life researchers to have preceded DNA in early life's history, forming what scientists call the RNA world hypothesis, in which RNA both carried genetic information and catalyzed its own chemical reactions before the more stable DNA system evolved. The presence of RNA backbone sugars in a 4.5 billion-year-old asteroid is not proof of that hypothesis, but it is consistent with it in a way that was not experimentally confirmed before the Bennu sample existed. Then came the ammonia. The Bennu samples contained exceptional concentrations of ammonia, significantly higher than anything detected in the Japanese Hayabusa 2 mission samples from asteroid Ryugu, and higher than in most meteorites. Ammonia matters because it reacts with formaldehyde, which was also detected in the Bennu samples, to initiate a cascade of chemical reactions known as Strecker synthesis, one of the primary abiotic pathways for amino acid formation. In the interior of Bennu's parent body, where liquid water moved through ammonia-rich, carbon-laden mineral matrices at an alkaline pH, Strecker synthesis would have been operating continuously. The asteroid was not just a passive carrier of molecules. It was, in a sense, a slow-moving chemical reactor, quietly manufacturing the precursors of life over millions of years in the dark of the early solar system. And then, in February 2026, a study published in the Proceedings of the National Academy of Sciences added another layer entirely. Researchers led by Penn State University examined the glycine found in Bennu, the simplest and most abundant amino acid in the sample, and found evidence suggesting it had not formed through the Strecker aqueous pathway at all. Instead, the isotopic signature of Bennu's glycine pointed toward a different formation route: synthesis in frozen ices exposed to ionizing radiation in the cold outer regions of the early solar system, or in the interstellar medium itself, before the solar system even fully assembled. The implication is significant. If confirmed, it would mean that some of Bennu's organic molecules did not form inside the asteroid during its wet geological history. They arrived at the asteroid already assembled, carried in ice-grain mantles from interstellar space, inherited directly from the molecular cloud that became the solar nebula. These molecules would predate the sun. They would be older than the solar system itself, older than the sun. The scientists who built the clean rooms, who designed the nitrogen purge containers, who spent years preparing to receive 121 grams of black gravel, they were not just studying the solar system. They were reading a molecular archive that may extend back into the interstellar environment that preceded it. And the further back that archive reaches, the more it forces a fundamental question about the uniqueness of life's chemistry. If these molecules form in interstellar space, they are not rare products of one particular solar system. They are a universal output of the physics and chemistry that governs all of space. Which raises a question that the next layer of analysis is only beginning to ask. If these molecules are everywhere, if the chemical ingredients of life are a standard product of the universe's chemistry, then what exactly is the bottleneck? What is the step that separates a rock full of amino acids and nucleobases from a living cell? And is that step something that happened once, by extraordinary accident, on one pale blue planet? Or is it something that the universe, with its billions of galaxies and its trillions of carbonaceous bodies, performs over and over again, in countless places, in the cold dark between the stars? Bennu did not form where it orbits today. Its current path around the Sun, a slightly elliptical orbit that crosses Earth's orbital path twice per revolution, is the product of billions of years of gravitational perturbation, collision history, and a subtle but relentless force that most people have never heard of. Bennu began its existence as part of a much larger object, a parent body that formed in the outer reaches of the main asteroid belt between Mars and Jupiter in the first few million years of the solar system's existence. That parent body was not a cold, inert rock. It was chemically active, it was warm, and the source of that warmth is the first piece of physics that explains everything that came after. In the earliest phase of the solar system, the young sun was surrounded by a disk of gas and dust, the protoplanetary disk, and within that disk, solid particles were colliding and clumping together under their own gravity in a process called accretion. The parent body of Bennu formed through this process, but as it grew, something happened inside it that transformed its chemistry entirely. The decay of short-lived radioactive isotopes, primarily aluminum-26 and iron-60, which had been scattered through the solar nebula by a nearby supernova shortly before the solar system's formation. Aluminum-26 has a half-life of approximately 717,000 years. That is brief on a geological timescale, brief enough that within the first few million years of the solar system's existence, enormous quantities of it were decaying inside newly formed rocky bodies, releasing gamma radiation and heat directly into the surrounding mineral matrix. The interior of Bennu's parent body, insulated by its own mass, began to warm from within. Not to the temperatures of volcanic activity on Earth, nowhere near that, but warm enough in the range of 50 to 150 degrees Celsius in the most active zones to melt the ice that had been incorporated into the body during accretion. That meltwater did not just sit there, it moved. Water percolated through the pore spaces between mineral grains, driven by pressure and temperature gradients, dissolving minerals as it traveled, precipitating new ones as conditions changed. This process, aqueous alteration, is the same chemistry responsible for the phyllosilicates detected in the Bennu sample. Dry and hydrous silicate minerals reacted with liquid water to produce hydrated clays. Carbon-bearing compounds dissolved, reacted, recombined. Ammonia, present in the original ice, entered solution. Simple organic molecules underwent condensation reactions in the alkaline, ammonia-rich water, building into more complex structures through chemistry that required no biological catalyst, only time, temperature, and the right molecular ingredients. The duration of this aqueous alteration phase on Bennu's parent body is estimated at somewhere between 1 million and 10 million years. That range is wide, and scientists continue to refine it. But even at the low end, 1 million years of continuous liquid water chemistry operating inside a carbonaceous body, the scale of molecular synthesis that could occur is almost incomprehensible. A single milliliter of liquid water contains approximately 33 billion trillion molecules. Every one of those molecules, in an environment rich in carbon, nitrogen, ammonia, phosphorus, and dissolved minerals, is a potential participant in a chemical reaction. Over 1 million years, across the entire interior volume of a body that was originally perhaps 100 kilometers in diameter, the number of individual chemical events that occurred is beyond any number that human intuition can meaningfully process. Think of it this way. If you assigned 1 second of your life to each molecular collision occurring in a single glass of water inside Bennu's parent body over just 1 year of aqueous alteration, you would be counting for longer than the current age of the universe. That is the scale of chemistry the Bennu data represents. The parent body eventually fragmented, almost certainly as the result of a catastrophic collision with another large object in the main asteroid belt, sometime in the range of 800 million to 1 billion years ago. The collision scattered debris across a wide arc of the inner solar system. Bennu is a remnant of that shattered interior, a loosely consolidated rubble pile held together not by a solid internal structure, but almost entirely by the weak self-gravity of its own mass. This is why its surface is so chaotic. Boulders ranging from a few centimeters to tens of meters, loosely stacked, occasionally tumbling in response to thermal forces. Bennu has no solid bedrock in the terrestrial sense. If you tried to land a spacecraft with significant mass on its surface, you would likely sink slowly into the material, like stepping onto a deep pile of very coarse gravel. The force responsible for Bennu's migration from the main asteroid belt to its current Earth-crossing orbit is called the Yarkovsky effect, and it is one of the most elegant examples of physics operating at scales too slow and too subtle for direct human observation. Every surface on Bennu absorbs sunlight during its rotation and re-emits that energy as thermal infrared radiation, heat. Because Bennu rotates, the side facing the sun absorbs energy, rotates away, and then re-emits that energy as the surface cools. The re-emission is not perfectly isotropic. It is slightly offset from the exact direction back toward the sun because of the lag between absorption and emission. That asymmetric re-radiation produces a tiny but continuous thrust. Tiny, in this case, means a force equivalent to the weight of a single grape acting on an object massing 73 billion kilograms. Over millions of years, that force adds up to a measurable change in orbital velocity. Over hundreds of millions of years, it is enough to shift an orbit from the main belt into the inner solar system, across the orbital paths of Mars and Earth. The Yarkovsky effect was first theorized in the early 20th century and remained largely academic until precise radar measurements of Bennu's orbital trajectory confirmed it was drifting at a rate of approximately 284 meters per year, exactly consistent with Yarkovsky predictions. This was not a minor confirmation. It was the first direct measurement of the Yarkovsky effect on a specific asteroid, and it gave scientists a tool to retrodict Bennu's past orbital history and predict its future trajectory with far greater precision than was previously possible. Those calculations are what produced the current impact probability figures and the specific date ranges of potential Earth encounter in the 22nd and 23rd centuries. The physical structure of Bennu itself tells the story of its violent history in layers. The surface material, the regolith, is predominantly made of fragments no larger than a few centimeters, produced by billions of years of micrometeorite impacts grinding the surface rock into finer and finer particles. Beneath that surface layer, larger boulders retain more of the original parent body material, less processed by surface irradiation and impact gardening. The largest boulder on Bennu's surface, informally called Bennu's boulder, is approximately 58 meters wide. On Earth, that would be roughly the length of an 18-story building lying on its side. On Bennu, it sits on the surface like a loose stone in a pile of gravel, held in place by gravity so weak that a strong throw from a standing human could launch a baseball into a stable orbit around the asteroid. The interior of Bennu presents its own scientific puzzle. Radar observations conducted by OSIRIS-REx during its orbital survey phase suggest that Bennu may be partially hollow, or at minimum, that its interior is significantly less dense than its surface rocks. The current best estimate places Bennu's bulk density at approximately 1,190 kilograms per cubic meter. For comparison, solid granite has a density of approximately 2,700 kilograms per cubic meter. Water is 1,000 kilograms per cubic meter. Bennu, in terms of bulk density, is barely denser than water, meaning that if you could find an ocean large enough to hold it, Bennu would very nearly float. This low bulk density is consistent with a rubble pile structure in which 30 to 40% of Bennu's interior volume is empty space, voids between loosely packed boulders and gravel. The material that fills the rest of that volume is not uniform. Near-infrared spectroscopy from orbit identified at least 3 distinct spectral units on Bennu's surface, suggesting that the original parent body may have contained distinct compositional zones that were scrambled and reassembled during the collision that created Bennu. Some boulders on the surface have spectral signatures significantly different from the surrounding material. They may be fragments of a completely different impactor object, incorporated into Bennu's rubble pile at the time of the collision, meaning that Bennu carries within it not only the chemistry of its own parent body, but potentially the chemistry of the object that destroyed it. The Nightingale crater, the sample collection site, is particularly significant in this context. It sits in the northern hemisphere of Bennu and is geologically young by asteroid standards, meaning its surface material has been exposed to space weathering, solar wind ion bombardment, and cosmic ray irradiation for a shorter time than the surrounding terrain. The fresher the surface, the less the original chemical signatures have been altered by space weathering, and the more representative the material is of Bennu's interior chemistry. OSIRIS-REx's choice of Nightingale was not arbitrary. It was the product of almost 2 years of surface spectroscopy, compositional mapping, and thermal inertia measurements designed to identify the site on Bennu's surface most likely to yield the least altered, most scientifically valuable material. Space weathering is its own physics lesson. On Earth, a rock sitting on the surface is protected by the atmosphere and the magnetic field from the direct bombardment of the solar wind, a continuous stream of charged particles, primarily protons and electrons, flowing outward from the sun at 400 to 800 kilometers per second. On Bennu, there is no atmosphere and no magnetic field. Every square centimeter of exposed surface is continuously bombarded by this particle flux, along with ultraviolet radiation and galactic cosmic rays. Over millions of years, this bombardment modifies the top few nanometers of mineral grain surfaces, altering their spectral reflectance and partially breaking down organic compounds. The material collected from Nightingale, while not immune to this process, had experienced less of it than most of Bennu's surface, which is precisely why the organic inventory it contained was so unusually rich. Organic chemistry in space does not require protection from radiation to survive. It can even be driven by radiation, as the detection of interstellar glycine forming in irradiated ice mantles demonstrates. But the layered, complex organics found in Bennu's sample, including the intact nucleobases and amino acid mixtures, were best preserved in material that had spent much of its history shielded within the parent body's interior, protected by overlying rock from the most destructive radiation fluxes. The aqueous alteration environment, warm, liquid, mineral-rich, enclosed, acted as both a chemical reactor and a protective matrix, synthesizing complex molecules and then locking them into the mineral structure of the clay minerals that crystallized as the water slowly froze or evaporated. Magnesium-sodium phosphate was also detected in the Bennu sample. Phosphate is the third element in the molecular backbone of both DNA and RNA, alongside carbon and nitrogen-bearing sugars and nucleobases. Its presence in Bennu, in a chemically available water-soluble form, means that all 3 backbone elements of genetic molecules were present simultaneously in the same aqueous environment inside this asteroid, not in separate locations, not under different conditions, together, in solution, in contact with each other, for millions of years. The chemistry was not performing a rehearsal. It was running the actual experiment at full scale, in the dark, 4.5 billion years ago. The results of that experiment arrived in Utah on September 24, 2023, in a capsule the size of a kitchen pot. And the most disorienting finding is still ahead. Because when scientists began comparing Bennu's chemical inventory not just to life on Earth, but to the other samples that humanity has managed to retrieve from the solar system, they found something that the models had not predicted: a discrepancy, a signature in the isotopic ratios that pointed not inward, toward solar system chemistry, but outward, toward something older, colder, and far more distant than any asteroid. The isotopes do not lie. That is not a philosophical statement. It is a precise description of how nuclear physics works. Every element on the periodic table exists in multiple forms, isotopes, that share the same number of protons but differ in the number of neutrons in their nucleus. Carbon-12 and carbon-13 are both carbon. Nitrogen-14 and nitrogen-15 are both nitrogen. Oxygen-16 and oxygen-17 and oxygen-18 are all oxygen. The ratio of these isotopes in any given material is not arbitrary. It is a direct record of the physical and chemical environment in which that material formed, a fingerprint written in nuclear structure that no amount of subsequent chemical processing can entirely erase. When the team analyzing Bennu's bulk elemental and isotopic composition published their findings in Nature Astronomy in August 2025, they were not reporting a minor refinement. They were reporting something that forced a fundamental revision of where Bennu's parent body formed in the solar system. And the answer pointed dramatically outward, past the orbit of Saturn, into the cold outer reaches of the protoplanetary disk. The key signal was in the nitrogen isotopes. Nitrogen-15 enrichments, meaning an excess of the heavier nitrogen-15 isotope relative to the lighter nitrogen-14, were detected throughout the soluble organic matter in the Bennu samples. The degree of enrichment was not subtle. It was pronounced, and it matched a pattern that planetary scientists recognized specifically from one class of environments: cold molecular clouds in interstellar space, or the extreme outer regions of the protoplanetary disk, where temperatures drop below 30 Kelvin, 30 degrees above absolute zero, and where chemistry occurs not in liquid water, but on the surfaces of ice-coated dust grains exposed to cosmic radiation. At temperatures that low, atoms and molecules do not have the thermal energy to move freely through a gas. They freeze onto the surfaces of dust grains, forming thin mantles of ice. In those ice mantles, exposed to ultraviolet photons and cosmic ray particles, chemistry still proceeds, but it is a different chemistry from the aqueous alteration that operated in Bennu's warm interior. It is a radiation-driven, low-temperature synthesis that preferentially incorporates the heavier nitrogen-15 into organic compounds because of quantum mechanical differences in the reaction rates of nitrogen-14 and nitrogen-15 at cryogenic temperatures. The process is called isotopic fractionation by ion-molecule reactions, and it leaves a nitrogen-15 enrichment signature that is essentially impossible to reproduce through any process operating at the higher temperatures found in the inner solar system. Finding that signature in Bennu means that a significant fraction of the nitrogen-bearing organic compounds in the sample did not form inside the asteroid during aqueous alteration. They arrived at the asteroid pre-assembled, carried in icy grain mantles from the interstellar medium or the outer protoplanetary disk, regions so cold and so distant from the sun that organic synthesis operated through an entirely different set of physical mechanisms. The same team identified pre-solar grains in the Bennu samples. These are among the rarest and most scientifically precious objects in cosmochemistry. Presolar grains are microscopic crystals, typically between 1/10 and a few micrometers in diameter, that condensed in the outflows of dying stars before the solar system formed. They are identifiable through their isotopic compositions, which are so radically different from solar system material that they cannot have formed within the solar system under any known process. When a massive star ends its life in a supernova, or when an asymptotic giant branch star expels its outer layers in a slow stellar wind, the cooling gas in those outflows begins to condense into solid grains: silicon carbide, graphite, oxide minerals, silicates. Each grain carries the specific isotopic signature of the nuclear reactions that occurred inside the star that produced it. In Bennu's samples, 7 oxygen-rich pre-solar grains were identified, including 2 silicate grains. The oxygen isotopic ratios measured in those grains, deviations from solar system baseline values of up to 8,067 parts per thousand in oxygen-17, are incompatible with any solar system formation process. These grains formed in other stars. They drifted through interstellar space, became incorporated into the molecular cloud that collapsed to form the solar nebula, survived the formation of the Sun and the protoplanetary disk without being destroyed by heat or shock, accreted into Bennu's parent body, survived the aqueous alteration phase, survived the catastrophic collision that shattered the parent body, survived billions of years of space weathering on Bennu's surface, survived the entry capsule's re-entry through Earth's atmosphere, and were recovered intact in a clean room in Houston. Each one of those survival steps represents an improbability. Together, they represent the kind of preservation that occurs only in the most chemically inert, low-temperature, primitive material in the solar system. The diversity of what survived inside Bennu was equally unexpected. The August 2025 Nature Astronomy study identified a rich mix of distinct material types within the samples: calcium-aluminum-rich inclusions, amoeboid olivine aggregates, and chondrule-like objects, alongside the fine-grained matrix that dominates the bulk of the material. Calcium-aluminum-rich inclusions, commonly abbreviated as CAIs, are the oldest dated solids in the solar system. Their age has been measured by uranium-lead radiometric dating at 4,567.3 million years old, with an uncertainty of plus or minus 0.16 million years. They are the first solids that condensed from the solar nebula as it began to cool, and they represent the time-zero reference point against which all other solar system chronology is measured. Finding them in Bennu, mixed with pre-solar grains that predate the solar system entirely, alongside interstellar organic matter, alongside aqueous alteration products that formed at much later times in the parent body's history, means that Bennu's parent body accreted an extraordinary range of materials from multiple locations and multiple time periods across the early solar system. This is the geological equivalent of finding a single rock that contains Precambrian basement material, ancient ocean sediment, volcanic ash from a separate eruption millions of years later, and grains of sand carried by wind from a different continent, all fused together into one sample. Bennu is a geological archive of the entire early solar system, not just the specific region where its parent body formed. The transport mechanism that explains how material from such different parts of the early solar system ended up in the same asteroid points to one of the most dramatic episodes in the solar system's early history: the Grand Tack and subsequent orbital instability events, during which the giant planets, Jupiter, Saturn, Uranus, and Neptune, underwent dramatic migrations through the protoplanetary disk. Jupiter's inward migration toward the Sun, followed by its reversal outward as it became caught in resonance with Saturn, scattered material from the outer disk inward and material from the inner disk outward in a massive dynamical mixing event. This churning redistributed solid bodies and their chemical contents across vast ranges of orbital distance, explaining why Bennu's parent body, which isotopic evidence suggests formed beyond Saturn's current orbit, contains high-temperature minerals like CAIs and chondrules that could only have condensed close to the young Sun. The data from Bennu's bulk isotopic composition, published in August 2025, went further still. The titanium isotopic ratios in Bennu were measured and found to be similar to those of the Ryugu asteroid samples returned by the Japanese Hayabusa 2 mission, and to the most primitive class of meteorites, the Ci chondrites. Titanium isotope ratios in solar system bodies vary systematically with formation location in the protoplanetary disk, and the Bennu value places its parent body's formation reservoir in the outer solar system. A conclusion consistent with the nitrogen-15 enrichments, the pre-solar grain abundances, and the elevated carbon and ammonia content that distinguish Bennu from inner solar system bodies. What the Ryugu comparison reveals is equally striking. Both Bennu and Ryugu are B-type and C-type carbonaceous asteroids respectively, and both were targeted by sample return missions specifically because of their primitive composition. Their organic inventories show broad similarities. Both contain amino acids, nucleobases, and complex soluble organic matter. But they differ in notable ways that reflect different degrees of aqueous alteration and different chemical environments in their respective parent bodies. Ryugu's samples are enriched in sulfur-bearing organic compounds and show higher ratios of beta-alanine to glycine, consistent with more extensive hydrothermal processing. Bennu's samples are richer in nitrogen-bearing compounds, show lower beta-alanine to glycine ratios, and contain 5 to 10 times higher abundances of nitrogen heterocycle compounds than Ryugu. The heterocycles are particularly significant. Many of the nucleobases detected in Bennu are N-heterocycles, meaning that Bennu's parent body appears to have been a more efficient producer of nucleobase-class molecules than Ryugu's, possibly because its aqueous alteration operated at lower temperatures and preserved more of the thermally labile nitrogen-rich organics that would have been degraded under hotter hydrothermal conditions. 2 asteroids, 2 sample return missions, 2 partially overlapping but distinctly different organic inventories, both rich in the molecular precursors of life, both shaped by water, carbon, and interstellar input, both now sitting in clean rooms on Earth, being taken apart molecule by molecule and isotope by isotope by the most precise analytical instruments in human science. In August 2025, the OSIRIS-REx team published a paper with a conclusion that stopped many readers mid-sentence. Based on the totality of the chemical and isotopic evidence, the pre-solar grains, the nitrogen-15 enrichments, the titanium isotope ratios, the high organic and volatile content, the analysis concluded that Bennu's parent body did not form in the main asteroid belt at all. It formed in the outer solar system, possibly beyond the current orbit of Saturn, in a region so cold and so far from the sun that its initial building blocks were predominantly ices, dust, and organic-rich interstellar grains. Only later, through the dynamical chaos of giant planet migration, was it transported inward to the main belt, where it spent billions of years before being shattered into the rubble pile that became Bennu. The molecular cloud that produced those interstellar grains is gone. It collapsed to form the sun and the solar system, dispersed into space, or was consumed by subsequent stellar formation. But the grains it produced survived. They are in Houston, in a nitrogen-purged container waiting for the next round of analysis. Somewhere in those grains is a direct chemical connection to a star that died before our sun was born. A star whose name we will never know, whose light reached no human eye, but whose nuclear furnace forged the atoms that eventually became part of the molecular architecture of life on Earth. The chain of custody runs from dying star to interstellar cloud to solar nebula to protoplanetary disk to outer solar system body to catastrophic collision to rubble pile to Earth-crossing orbit to Utah desert to Houston, clean room to mass spectrometer. Every link in that chain held. And what the spectrometer read, when it finally reached the end of the chain, was not a story of Earth's uniqueness. It was a story of cosmic continuity, of chemistry so universal, so robust, and so ancient that the molecules of life appear to be not an accident of one planet, but a standard output of the universe's most basic physical processes, distributed across space by the same gravitational forces that built the solar system in the first place. The question that remained, the one no spectrometer had yet answered, was whether those molecules, once delivered to a wet, warm planetary surface, reliably crossed the threshold into something that can replicate itself. And the search for that answer is now pointing scientists not backward toward the asteroid belt, but outward, toward the moons of Jupiter and Saturn, where liquid water oceans exist today, sealed beneath kilometers of ice, in the dark. Tryptophan. Of all the molecules that could have appeared in a 4.5 billion-year-old asteroid, tryptophan was among the last that scientists expected to find. It is one of the most structurally complex of the 20 protein-forming amino acids, a large aromatic molecule built around an indole ring system that requires multiple sequential chemical steps to synthesize even under controlled laboratory conditions. Meteorite science had searched for it for decades across thousands of samples. It had never been detected. Not once. Not in the Murchison meteorite, which fell in Australia in 1969 and has been studied more intensively than almost any other extraterrestrial rock in history. Not in the Allen Hills meteorites from Antarctica, not in the Tagish Lake carbonaceous chondrite, not in Ryugu. It was in Bennu. In November 2025, a study published in the Proceedings of the National Academy of Sciences reported the tentative but rigorously supported identification of tryptophan in the OSIRIS-REx sample, raising the total count of protein-forming amino acids detected in Bennu to 15 of 20. Jose Aponte, an astrochemist at NASA's Goddard Space Flight Center and a co-author of the study, stated directly, "The discovery of tryptophan in the Bennu asteroid is significant, as it is one of the more intricate amino acids and had not been observed in any meteorite or space sample until now. The reason tryptophan had never been found before, and the reason finding it in Bennu carries such weight, is precisely its complexity. Simple amino acids like glycine form through well-understood abiotic chemistry pathways that operate under a wide range of temperatures and conditions. Tryptophan does not. Its synthesis requires a specific sequence of reactions, a more complex molecular architecture, and conditions that allow those reactions to proceed through multiple intermediate stages without the intermediates being destroyed. Finding it in Bennu does not prove those conditions were met inside the asteroid. It proves they were met somewhere, in the interstellar medium, in the outer protoplanetary disk, in the cold chemistry of the ice grain mantles that fed Bennu's parent body, and that the molecule survived intact through 4.5 billion years of cosmic history to arrive in a Utah desert inside a heat shield. The survival alone is a story in physics. The re-entry capsule carrying the Bennu sample hit Earth's atmosphere at approximately 43,000 kilometers per hour. At that velocity, the compression of air ahead of the capsule generated temperatures of approximately 2,900 degrees Celsius on the heat shield's exterior surface, hot enough to vaporize steel, hot enough to ionize the surrounding air into plasma, hot enough that the capsule was briefly surrounded by a sheath of glowing superheated gas visible from the ground like a meteor trail. The sample inside survived because of a thermal protection system engineered specifically to keep the interior of the capsule below the temperature thresholds at which the most fragile organic compounds would degrade. The margin between the temperature on the outside of the heat shield and the temperature experienced by the sample inside, it was roughly 2,800 degrees Celsius. That margin was maintained for approximately 13 minutes, the duration of peak heating during atmospheric entry. The contrast in physical scales that this represents is almost architectural. On the heat shield's exterior, plasma at nearly 3,000 degrees. Inside the sample container, a sealed nitrogen atmosphere at near ambient temperature, preserving molecules that had survived in interstellar space since before the sun existed. Engineering protecting chemistry, protecting biology's oldest archive. When the capsule was opened at Johnson Space Center, the team found something that immediately told them the collection had exceeded expectations. The touch-and-go mechanism's sample collector head was so full of material that rocks had jammed the closure flap. Images taken by OSIRIS-REx's onboard camera immediately after collection showed dark material visibly escaping from the collector housing. Material drifting away in Bennu's microgravity, lost to space, never to be recovered. The sample that made it to Earth was the portion that stayed inside. Scientists estimate that a non-trivial fraction of the collected material was lost during those first minutes after collection, before the head could be sealed. What arrived in Houston, 121.6 grams, was the remainder. 121 grams that contained tryptophan. That contained all 5 nucleobases. That contained the evaporate sequence of 11 distinct salt minerals deposited by ancient brine. The evapораtes deserve particular attention because they represent one of the most complete mineral records of liquid water chemistry ever recovered from any extraterrestrial body. Evapораtes form when a mineral-saturated solution evaporates, leaving behind the dissolved salts in a specific sequence determined by their solubility. The order in which minerals crystallize out of an evaporating brine is a precise function of the brine's original chemistry, its pH, its ionic composition, its temperature. By reading the evaporate sequence in Bennu's samples, calcite, halite, sylvite, and then trona, which was detected in extraterrestrial material for the very first time, scientists at Lawrence Berkeley National Laboratory reconstructed not just the presence of liquid water on Bennu's parent body, but the specific chemistry of the water, the direction in which it evolved as it evaporated, and the approximate duration of the evaporation process, which the data suggests lasted thousands of years. Trona, sodium sesquicarbonate, forms at the very end of an alkaline evaporation sequence, after calcite and halite and the other more soluble salts have already precipitated. Its presence in Bennu means the evaporation sequence ran to completion. The water did not simply freeze or drain away before the chemistry finished. It evaporated fully in an alkaline, carbon-rich, ammonia-bearing environment over a timescale long enough for the most soluble minerals to crystallize last. That sequence, thousands of years of slow evaporation inside a warm, sealed, chemically productive environment deep inside a primitive asteroid, is exactly the kind of sustained aqueous chemistry that prebiotic chemistry models require to produce the concentrations of complex organics found alongside those evaporites. The Berkeley lab team used scanning transmission X-ray microscopy at the Advanced Light Source facility to analyze the Bennu material at the nanometer scale, imaging individual chemical bonds in individual grains of the asteroid, mapping the distribution of organic carbon, nitrogen, and oxygen at resolutions finer than the wavelength of visible light. At that scale, they found that the evaporate salts were not simply sitting on the surfaces of mineral grains. They were mixed into the rock at the finest compositional levels, intimately intergrown with the organic compounds and the clay minerals in a way that could only have formed if all three components were present simultaneously in the same aqueous solution. The salts, the organics, and the clays co-precipitated. They formed together from the same brine, in the same environment, at the same time. The implications of that co-precipitation are not abstract. One of the central challenges in origin of life chemistry is the concentration problem. How do dilute solutions of prebiotic molecules reach the concentrations necessary for the reactions that produce RNA, proteins, and ultimately self-replicating systems? In a vast ocean or a large body of water, organic molecules disperse. Their concentrations drop. Reaction rates fall. The chemistry stalls. Evaporation solves the concentration problem directly. It reduces the volume of the solution, increasing the concentration of every dissolved species simultaneously, forcing molecules that were too dilute to react in the open ocean into close proximity inside the shrinking brine. Bennu's parent body ran that experiment for thousands of years. In an enclosed rocky environment, with alkaline water, with amino acids in solution, with nucleobases in solution, with phosphate minerals dissolving into the brine, with ammonia providing nitrogen for further synthesis. The evaporate sequence in the Houston clean room is a frozen record of that experiment's final phase, the moment when the brine reached its most concentrated state and began to crystallize into the mineral sequence that OSIRIS-REx brought home. Whether the experiment produced anything more than its molecular ingredients, whether any of the organic chemistry inside Bennu's parent body crossed any threshold toward self-organization, toward catalysis, toward replication, is a question that the Bennu data cannot answer. The sample does not show evidence of life. It shows evidence of all the chemistry that, on Earth, preceded life. The distinction matters enormously, and serious scientists are careful about it. But the distinction also raises a question that the data forces into focus with unusual clarity. On Earth, we know that the gap between a warm, alkaline, organic-rich, concentrated aqueous environment and the first self-replicating molecule was crossed. Once. At least once, 4 billion years ago, in circumstances that the geological record has not preserved well enough to reconstruct in full. The question of whether that crossing is rare or common, whether it requires extraordinarily improbable conditions, or whether it follows almost inevitably from chemistry of the type documented in Bennu, is the deepest open question in all of science. The Bennu data does not resolve it, but it narrows it. If the organic chemistry documented in Bennu's samples is representative of carbonaceous asteroids generally, and there are thousands of them in the main belt alone, with millions more in the Kuiper belt and the scattered disk beyond Neptune, then the molecular feedstock for life is not rare in the solar system. It is abundant, it is distributed, it was delivered to the early Earth in quantity during the late heavy bombardment by objects whose chemistry resembled Bennu's. The probability calculation for the origin of life, whatever its current value, must be computed against a background of that molecular abundance, not against a background of a sterile, organic poor early Earth assembling its chemistry from scratch. The Curiosity rover on Mars has detected organic molecules in Gale crater sediments. The Cassini mission detected molecular hydrogen and organic compounds in the plumes erupting from Enceladus's subsurface ocean, an ocean that exists today in contact with a rocky seafloor, at temperatures and pressures broadly consistent with the hydrothermal environments where some origin of life models are set. The James Webb Space Telescope has detected dimethyl sulfide in the atmosphere of the exoplanet K2-18b, a molecule that on Earth is produced exclusively by marine phytoplankton, though abiotic formation pathways cannot be ruled out. None of these detections constitute evidence of life. Each of them, however, is consistent with chemistry that the Bennu data shows was operating at full scale across the solar system 4.5 billion years ago. The amino acids in Bennu are the same amino acids in your body. The nucleobases in Bennu are the same nucleobases in your DNA. The phosphate minerals in Bennu are chemically continuous with the phosphate backbone of every gene you have ever inherited. The water that flowed through Bennu's parent body and deposited those evaporates operated at the same alkaline pH as the hydrothermal vents that some researchers believe were the cradle of terrestrial life. These are not metaphors. They are chemical identities. The molecules are the same molecules. The physics that produced them in the outer solar system 4.5 billion years ago is the same physics that operates in every molecular cloud, around every young star, across every galaxy in the observable universe. 15 of the 20 amino acids of life. All 5 nucleobases. The backbone sugars of RNA, phosphate, ammonia. Alkaline water, concentrated by thousands of years of evaporation, mixed at the nanometer scale with organic compounds that formed in interstellar space before the sun was born. All of it in 121 grams of black gravel. All of it waiting in a nitrogen-purged container in Houston. All of it written, clearly, legibly, in the only language that does not lie, in the isotopic ratios of atoms that condensed in the outflows of stars that died before our solar system existed. The universe did not hide this. It preserved it. For 4.5 billion years, across 7 billion kilometers of interplanetary space, through the plasma of atmospheric re-entry, through a 6-second contact with a robotic arm in the microgravity of an asteroid's surface, it preserved it and delivered it, and waited for a species that had only just learned to read. The sample is still being opened. That is not a figure of speech. As of early 2026, the majority of the Bennu material collected by OSIRIS-REx has not yet been analyzed. NASA's curation policy for sample return missions mandates that a substantial fraction of any returned extraterrestrial sample be preserved unopened for future generations of scientists who will bring analytical techniques that do not yet exist. Approximately 70% of the Apollo lunar samples collected between 1969 and 1972 were sealed in their original containers and stored at Johnson Space Center, waiting decades for science to catch up to the material. Some of those samples were finally opened in the 2010s and 2020s, and they yielded discoveries, including the detection of water ice in permanently shadowed lunar craters consistent with hydrogen signals measured by Apollo samples. That would have been missed entirely if every gram had been consumed by the analytical methods of the 1970s. The Bennu archive will follow the same logic. Of the 121.6 grams returned in September 2023, approximately 4 grams had been allocated to the initial curation and analysis teams by early 2025. The remaining material, more than 95% of it, sits in hermetically sealed containers under nitrogen atmosphere at Johnson Space Center, waiting. Some of it will be analyzed by mass spectrometers that have not yet been built. Some of it will be interrogated by techniques that currently exist only in theoretical form. Some of it may be examined by scientists who have not yet been born. The molecules in those containers will still be there. They have already waited 4.5 billion years. A few more decades changes nothing about their chemistry. This patient preservation of scientific material is itself a statement about how knowledge accumulates. Not in sudden revelations, but in layers. Each generation of scientists reading deeper into the same archive, extracting meaning that was always there but invisible to instruments less precise than the ones that finally looked. The Bennu sample is the most complex extraterrestrial chemical archive ever held in human hands, and the science community's deliberate choice to ration access to it is an acknowledgement that the archive will outlast any single generation's ability to read it fully. What has been read so far is enough to reshape the field. Before OSIRIS-REx, the best chemical data on primitive carbonaceous material came from meteorites, rocks that had survived a fiery, uncontrolled descent through Earth's atmosphere landed in fields or deserts or oceans, and been exposed to terrestrial moisture, bacteria, handling, and contamination before reaching a laboratory. The Murchison meteorite, which fell in Victoria, Australia in September 1969, became the gold standard of primitive meteorite chemistry for more than 5 decades. It contained over 90 amino acids, including many not found in biology. It contained nucleobases. It contained complex polycyclic aromatic hydrocarbons. For 50 years, it was the richest organic inventory ever measured in an extraterrestrial sample. Bennu, in 121 grams of carefully curated nitrogen-sealed contamination-controlled material, exceeded it. Not marginally, substantially. The richness and complexity of the organic inventory, the preservation of labile molecules like tryptophan that terrestrial contamination and atmospheric heating would have destroyed in any uncontrolled fall, and the presence of minerals, trona among others, never previously detected in extraterrestrial material, collectively established that meteorites, for all their scientific value, had been giving the scientific community a systematically degraded picture of what primitive, carbonaceous material actually contains. Every meteorite that has ever been studied arrived damaged. Bennu arrived intact. The implications cascade outward from that single fact. If the Murchison meteorite's organic inventory, already extraordinary by any measure, represents a degraded version of what its parent body originally contained, then the molecular richness of carbonaceous asteroids in the solar system has been consistently underestimated for 50 years. The models of prebiotic chemistry built on meteorite data are models built from incomplete inputs. The calculations of how much organic material was delivered to the early Earth during the late heavy bombardment need to be revised upward. The probability estimates for the emergence of life, insofar as they depend on the availability of molecular precursors, rest on a foundation that was systematically too conservative. None of this proves that life is common in the universe. Science does not operate by proof in that direction. It operates by probability revision, by the progressive narrowing of uncertainty as new data constrains the parameter space. What Bennu has done is shift the probability distribution. The question is no longer whether the molecular ingredients of life are available on primitive solar system bodies. They are. The question shifts to the next step. Whether the conditions required to organize those ingredients into self-replicating systems are also widespread, or whether they represent a far rarer bottleneck. That question is now being pursued simultaneously across multiple missions and multiple worlds. The Europa Clipper spacecraft, launched in October 2024, is currently en route to Jupiter's moon Europa, which harbors a saltwater ocean beneath approximately 15 to 25 kilometers of ice. That ocean is in contact with a rocky seafloor, heated by tidal forces from Jupiter's gravity, and may contain hydrothermal vents operating on chemistry broadly analogous to the alkaline hydrothermal vent environments that some origin of life models favor. The Clipper will not be able to drill through the ice, but it will be able to characterize the composition of Europa's surface, which is periodically refreshed by material from the ocean below, and to search for organic molecules, salts, and potential biosignatures in the thin material ejected into Europa's tenuous exosphere. Enceladus, Saturn's small, brilliantly white moon, is already known to be actively venting its subsurface ocean directly into space through the tiger stripe fractures at its south pole. The Cassini mission, which ended its operational life in September 2017 by deliberately diving into Saturn's atmosphere to avoid contaminating Enceladus, detected molecular hydrogen, silica nanoparticles, carbon dioxide, methane, and complex organic molecules, including compounds with molecular masses above 200 atomic mass units, indicating large, structurally complex organic structures in those plumes. A future dedicated Enceladus mission, currently in the proposal and study phase, could fly through those plumes and collect samples without a spacecraft ever needing to land. The chemistry is already in space, waiting to be caught, delivered by the moon itself. On Mars, the Perseverance rover has been drilling core samples from the ancient lakebed sediments of Jezero Crater, a site where a river delta once deposited sediment into a lake that existed approximately 3.5 billion years ago, at a time when Mars had liquid water at its surface. Those cores are sealed inside titanium tubes and cached on the Martian surface, awaiting a future sample return mission. Whether they contain organic material, and if so, what kind, will not be known until those tubes reach a laboratory on Earth. When they do, the comparison with Bennu's organic inventory will become one of the most scientifically productive datasets in the history of planetary science. Two primitive bodies, one that stayed cold and dry, one that was warm and wet, both analyzed at the same level of instrumental precision, both read against the same molecular reference library that Bennu has now established. The thread that runs through all of it, through Bennu's amino acids and nucleobases, through Europa's hidden ocean, through Enceladus's active plumes, through the cached cylinders on the floor of Jezero crater, is water. Liquid water, sustained over geologically meaningful timescales, in contact with rock, in the presence of carbon and nitrogen and phosphorus. Everywhere that combination appears in the solar system, scientists now know that the molecular precursors to life are likely to be present as well, because Bennu has demonstrated that those precursors are a standard product of the chemistry that operates in carbon-rich, water-bearing planetary bodies. The Bennu sample also quietly answered a question that had persisted at the edges of origin of life research for decades. The chicken and egg problem of genetic molecules and metabolism. RNA, which can both carry genetic information and catalyze chemical reactions, is the leading candidate for the first self-replicating molecule in the RNA world hypothesis. But RNA requires ribose, a 5-carbon sugar, as its backbone. Ribose is notoriously unstable under many prebiotic chemistry conditions. Its detection in Bennu, alongside the nucleobases that attach to it, and alongside the phosphate minerals that complete the RNA backbone, means that all 3 molecular components of RNA were present simultaneously in the same extraterrestrial aqueous environment. Not in separate experiments, not in separate locations, not under hypothetical conditions, but together in real material preserved for 4.5 billion years. The RNA world is no longer purely theoretical. It now has a plausible materials supplier. That supplier was an asteroid in the outer solar system that formed before the Sun reached its current size, accreted chemistry from interstellar grains that predated the solar system entirely, was warmed from inside by the decay of radioactive aluminum, experienced millions of years of liquid water chemistry in its interior, was shattered by a catastrophic collision, drifted inward through billions of years of gravitational perturbation and Yarkovsky drift, crossed the orbit of Earth, was intercepted by a spacecraft from a civilization that had existed for roughly 300,000 years out of the solar system's 4,500 million, and was brought home in a capsule that landed in the Utah desert on a Tuesday morning in September. The Tuesday morning is not incidental. It is a timestamp in a chain of custody that stretches back to the interstellar medium, to conditions that existed before there was a sun to orbit, before there was a solar system to belong to, before there was an Earth to receive what the universe had been preparing. Sarah Russell, cosmic mineralogist at the Natural History Museum in London and co-lead author of the January 2025 Nature paper, said, "There were things in the samples that completely blew us away. The richness of the molecules and minerals preserved are unlike any extraterrestrial samples studied before." That statement came from a scientist who has spent her career studying meteorites. She was not describing excitement at confirmation. She was describing genuine surprise. The particular quality of surprise that only occurs when reality exceeds the most optimistic version of what was expected. Science operates best in that register. Not in the confirmation of what was already believed, but in the discovery that the universe is more generous than the models predicted. Bennu was more generous. Its chemistry was richer, more complex, more complete, more ancient, and more directly connected to the molecular architecture of life than any meteorite had suggested was possible in a single sample. What it means is still being worked out. The papers are still being written. The allocations of remaining sample material are still being planned. The instruments that will read the next layer of the archive are still being built. The missions that will carry Bennu's molecular legacy to other worlds, to Europa, to Enceladus, eventually to the sample tubes on the floor of Jezero crater, are still in transit, still in development, still in the minds of scientists who read the Bennu results and began asking the next question. The universe is approximately 13.8 billion years old. It has been producing the molecular ingredients of life, in molecular clouds, in protoplanetary disks, in carbonaceous asteroids, in the icy mantles of interstellar dust grains, for most of that time. The evidence now sits in Houston, in 95% of an unopened archive, in nitrogen-purged containers, at room temperature, patient and complete. 4.5 billion years is how long it took for the molecules to find an instrument precise enough to read them. The reading has barely begun.